nucleic acid to polypeptide, which is a chain of amino acid. We'll answer these questions a bit. Here is the clover leaf looking tRNA. You can see at the top there's an amino acid, and on the bottom there is an anticodon, which corresponds to what's on the mRNA, which is at the ribosome. There are a distinctive clover leaf. You have the anticodon, which is 3 nucleotides long, which matches with what's on the mRNA. Going back to the concept of the wobble hypothesis, you can see on the right, you have the UAC, which would be on the mRNA, and that could code for AUA, which would code for tyrosine, and on the right, if you had UAU instead of UAC, that would do the same thing. So we talked about this before, the wobble hypothesis, and where you can get that redundancy built in to correct for mutations. You can see when we're reading, we automatically and mentally autocorrect, so we can still read it and it makes sense. I had 2 errors there, but you could still see the problem. Because of the wobble hypothesis, you don't actually need 61 different tRNAs, because one tRNA can code for a few different things, but it still only picks up one amino acid. The process of adding an amino acid to tRNA is called amino acylation, or it's called charging a tRNA. And this process is catalyzed by a specific enzyme, and their energy comes from the complex itself. Let's look at the ribosome. You can see there's the A, P, and E site. So on the right, there's the A site, where the tRNA is coming in with the amino acid, the P site, or the peptidyl site, where the amino acids are being linked together, and the E site, where the empty tRNA is leaving on the left. Ribosomes use mRNA like an information tape, and it's red, and the information is used to join the amino acids into a polypeptide sequence. You can look at those later. There's a special binding site for the mRNA to come together. I already mentioned the A, P, and E sites. So there's 3 parts to translation. The first is initiation, the second is elongation, and the third is, I can't remember, something that means stop. So in the first step, you have the tRNA attached to methionine, attaching to the small ribosome subunit. Then these 2 things grab onto the 5' cap of the mRNA and zoom along. And then the last part happens, which is the large ribosomal subunit attaches. I like to think of it like a hamburger, where there's a bottom bun, and which is a small ribosomal unit, and then the information tape, or the meat, which is like the mRNA, and then the large subunit. Okay, then we look at elongation. So the first thing that happens is the tRNA with the amino acid on it, with the second amino acid on it, goes into the A site, and then the peptidyl transferase, or that enzyme, will help attach the second amino acid to the first one. And then the ribosome moves along the mRNA, and the empty mRNA is released. Empty tRNA is released. Let me just go back and make sure I didn't forget anything. The initiator tRNA pairs with the AUG. Okay, so there is a correct reading frame which starts with the AUG, which is the start codon, and the AUG starts the reading frame, and then as the ribosome goes along, it reads 3 base pairs at a time. Sorry, 1 codon at a time, which is 3 bases. GTP is used for this step. Step number 2. I think that's all I missed. All right. Okay, so going back a bit, the tRNA comes into the A site, then it moves along, the amino acid attaches to the other amino acid, which is in the P site. The amino acid moves along to the next codon in the reading frame, and the empty tRNA goes out the E site. So the last stage of translation, so the first was initiation, second was elongation, and the third was termination. So we talked about this earlier, but when one of the stop codons arrive in the A site, then translation stops. And then protein release factor binds at this site instead of another amino acyl tRNA, and then everything separates. At this point, what you have is a string of amino acids called polypeptide, but it's inactive, so it cannot be used. If it's an enzyme, it can't work. What has to happen is it has to have the primary, secondary, tertiary, quaternary structures in order for activation to occur. And that's it. Hope you enjoyed the lesson.